So in our last video, we went over the steps of transcription and translation. And so for this video, we're going to talk about some detail-oriented things describing how all of this works together. We're going to cover the base pair rules of RNA, and we're going to cover the codon chart, which tells us what RNA codons translate into for proteins. So remember, transcription is where we take the DNA strand, that's this thing here in blue, and we're going to be making an RNA strand by the RNA nucleotides bonding to these bases that are revealed as we unzip the DNA. So the base pair rules for RNA are that A bonds to U and C bonds to G. So adenine now bonds with uracil, cytosine still bonds with guanine. So we no longer have thiamine to use. So now that we have uracil, it's going to take the place of thiamine. And then we talked about translation, where we have the RNA here at the bottom of the tRNA, that anticodon, that sequence on the tRNA that bonds to the sequence here on the mRNA. It also has an amino acid at the top. And when the anticodon bonds to the codon here in this binding site, we're going to connect the amino acid chain to this amino acid up here and continue to build the chain, as you can see in this picture. In these two ribosomes down here, you can see that amino acid chain being built. So let's practice using the base pair rules of RNA. If we have a DNA sequence, ATC, GTT, CGA, ACG, then the RNA sequence that would bond to it when we're building our mRNA strand in transcription would be as follows. We would put U bonding to A. So we're going to form a bond here between these two bases. Remember, A bonds to U. So anytime that we would see something like a T, we're going to have to use the letter U for uracil instead. So here the RNA sequence would be U bonding to the A. And then here's where it gets a little tricky. When we're bonding to DNA, A will still bond with T. So we can still use A in place of U here when we're building the mRNA strand. Remember, the base pair rules just say what bonds with what. RNA can only use U, A, C, and G. So when we're constructing our first strand of mRNA, we have to follow those base pair rules. U will bond anytime that we have an A in the sequence. A will bond anytime we have a T in the sequence. So our sequence would be UAG, CAA, GCU, UGC. And we follow those base pair rules. Now the anticodon, the part of the RNA that would be found on a tRNA, is going to bond to the strand of mRNA. So we're going to just use straight RNA base pair rules here. So anytime we'd have a U in the sequence, what's going to bond to it is an A. So our sequence here is UAUC. So we have A bonding to U, we have U bonding to A, and C bonding to G. And one of the things you might notice is the anticodon sequence looks pretty similar to the DNA sequence, except it uses U instead of T. This is something we're going to practice in class multiple times. So if you're a little bit worried about how to do this, you might take some time to stop and practice this yourself, following the base pair rules in order to make sure you understand how this sequence works. So now that we've talked about the base pair rules that govern transcription and translation, let's talk about how we get to proteins. So remember, we have our strand of mRNA, and that strand of mRNA is what is going to give us the instructions to build our protein. And we build it from the tRNAs that have the anticodon that can read the sequence of mRNA and that amino acid at the top. So we, as biologists, have figured out what sequences of nitrogen-containing bases code for which amino acids. So this is a codon chart, and it's very straightforward to use. We take the first base in the sequence. Let's say it's A. We're going to look for the first base in our sequence. It's not a U, it's not a C, but here it is with A. We know we're going to be in this row of boxes. Then let's say we have our second base is a G. So we come to the top side of this chart, we look for the letter G, and we know we're going to be in this box right here. Then we're going to look for our third base. Let's say it's C. So our codon for this case was A, G, C. So we know we're in this row of boxes, we know we're in this column of boxes, and we're going to look for the correct line, which is going to be this C line right here with AGC. So we found the right codon, and then we have our amino acid here next to it. It has the full name, serine, and it has the 3-letter abbreviation SER. This is how we can find all of the amino acids. So when we look at this sequence of mRNA, we can actually read it and convert each individual codon into a strand of amino acids. Again, this is something we're going to be practicing in class. So this is something that is kind of easier to do rather than just listen to. So don't worry, you are going to have some practice with this. So it's just a reminder about the central dogma, this idea of the flow of information. Remember, it flows from DNA to RNA to proteins. And when we're talking about taking DNA to RNA, that process is called transcription. When we're taking RNA to protein, that is a process called translation. Both transcription and translation are parts of protein synthesis, so making proteins. We can also include DNA replication in this process. That's what this blue arrow here is. That's just showing you how DNA can be replicated and stay inside the nucleus. It doesn't go anywhere. So that's what that is showing you here. That is what we're going to cover with protein synthesis. We're going to be working with these ideas in class as well, so you will get practice with some of these ideas.